In the past several years, I have been using electron microscopy and advanced technique to study various superstructures, interaction between nanoparticle, and correlate the structure property relationship. I like to represent the evolution of microscope and microscopy in the form of a tree because today microscope has branched into different discipline where it has given enormous amount of advanced scientific knowledge in various branches of science. At the same time, various branches of sciences have also contributed to the development of microscope. If you look at in the 19th century, after up-based diffraction limit, and today we can overcome this limit using various methods and technology. If you have noticed August 21st issue of journal Science, we come across what we call as attosecond microscopy or automicroscopy. Today, microscope is not used simply magnifying an object. Instead, today we can monitor the reaction real time. We can visualize atoms in 3 dimensions. We can solve the structure of biological molecule at atomic scale resolution. And more importantly, it is possible to monitor real-time synthesis of nanoparticles, understand the chemistry of battery material. You can do biasing and electrochemistry using nano to picoliter volume of samples. That is the power of transmission electron microscope today, which I'm going to talk. And I told about microscopy has evolved over the years and various fields have contributed. Let me give an example of a tomography where Radon has first proposed the mathematical formulation for constructing the three-dimensional volume using 2D projections. In 1931, Ansruska and al. Built the first transmission electron microscope, and after a few years, Turkevich and co-workers utilized transmission electron microscopy to study various forms of gold source. If you read the 1949 and 51 papers by Turkevich and co-workers, you will see extensive study of using transmission electron microscope to understand the size and shape and the effect of various reaction and solvents in gold nanoparticles. More importantly, at the same time, biologists have become more interested in discovery of DNA and studying various plant virus particles. Crane HR proposed that viruses are a great example for subassembly. Watson and Craig mentioned and proposed that plant virus is a molecule we can consider today as a colloidal molecule because it has a very well organized and ordered diffraction pattern. However, transmission electron microscopy tomography was first realized in 1968 by De Rozier and Aaron Klug, and they used a three-dimensional reconstruction using 2D projections of plant virus particles. I have been using transmission electron microscopy and tomography in the past 10 years to study a variety of self-assembled structures, including nanoclusters, nanoparticles. It can be toroid-like structures, spherical particles, looking at their internal structure, and even UFO-like structures, and looking at the diversity and heterogeneity within its structures. We have looked at virus nanoparticles, and we looked at superstructures and their internal structure, able to understand the position orientation of individual nanoparticle, which allow us to collect the data, information rich data for simulation and provide data for predictive design and engineering of nanomaterials. This work has resulted in a tremendous amount of more advanced knowledge and scientific output. Using this tremendous amount of information, in 2023, I also proposed that today very well-defined nanoparticles such as atomically precise nanoclusters can be used as an analogy to virus capsid formation. And I propose that it's possible to engineer precisely what we call as a precision nanoengineering across length scales. Let us take an example of virus particle here. And we can look back and look at the individual subunits of this. A simple CCMV like virus has three different subunits which can form pentamer or a hexamer and which eventually result in a capsid containing a definite number of pentamer and hexamers. And we can today crystallize routinely and look at the three-dimensional structures. If you take nanoparticles and nanocluster, they have very well organized and atomically precise and exact number of metal atoms inside and a ligand shell. And if you look at the three-dimensional structure and surface view of a nanocluster, it exactly looks like a highly organized, patchy ligand distributed surface. So we can consider them as a rigid virus-like particle with a patchy distribution of ligand bundles. By precisely controlling the interaction between the ligands, today we can make three-dimensional crystals, 2D colloidal crystal, macroscopic membrane, toroid-like structure, capsid. Therefore, I like to say that we are not only able to make hierarchical and complex structure, but using nanoparticle, we can make very structurally diverse materials. And is it possible? Yes, we have recently demonstrated that we can make large scale membrane which is monolayer thick and each membrane has one nanoparticle thick and they can behave same as polymer membranes purely based on the hydrogen bonding interaction between nanoparticles and with Young's modulus up to 15 gigapascal. However, today I have a different hypothesis. The hypothesis is that all materials undergo changes under certain environmental conditions. These changes will result in altered performance of devices or activities or catalysis. However, today we rely on understanding these structural changes under certain environmental conditions by imaging static pre or post structural transformation, which provides limited information, and very often it is not even possible to use these materials to image after once it is used. Cryo-electron microscopy can be used to arrest and freeze the certain stage during the transition. Here is an example of a steroid molecule that forms first micelles, and after 4 days it forms two-dimensional sheets. After 28 days it forms helical structures. However, we are facing multiple challenges. One of the challenges of nanomaterials under extreme condition is that visualization under real time. Can we understand the nanoscale dynamics? Can we study the atomic rearrangements? And this kind of image requires high spatial and temporal resolution. And when I say spatial resolution, we look at the atomic level resolution. In temporal, it can be anywhere between picosecond to seconds or hours. So if we are able to do that, we could be able to do a design optimization of functional materials for advanced technological application in extreme conditions. We can study many such things using cryo-EM today, but they are limited. It's not a real time. These are frozen structures at a definite time interval, which is tedious. When we talk about nanoscale dynamics, there are multiple types of dynamics. We can talk about vibrational dynamics, charge transfer, phase transition, or single particle dynamics. And all these dynamics can occur upon applying certain stimuli or trigger. For example, if you use a light and magnetic field, we are able to look at the vibrational dynamics. More importantly, these dynamics occur at a different time scale. Particularly, these can be imaged provided we have a different kind of technical advances integrated to transmission electron microscopy. What I'm really interested is this kind of triggers using electrons. So we can broadly divide this kind of nanoscale dynamics into two types. The one which is the transport of energy, charge, spin, and or quasi-particles, which lies in the range of femtoseconds to microsecond length scale, time scale. We can also look at structural reorganization, which is topic of interest for me. and it involves atomic diffusion or nucleation barrier, which occurs in milliseconds, seconds, or even hours. So when I talk about structural reorganization using electron beam, we have to understand that all materials interact with the electron beam because electron beam dose is an high energy radiation, especially when we use high dose. What happens when a beam of electron interact with your material? There one possibility is an elastic collision, which leads to the diffusion, atom displacement, or vacancy creation. Or under inelastic interaction, it will lead to electronic excitation, radical formation, ion fragments, increase in the temperature, contamination of your sample or the microscope. Then you can ask, what is a dose? A dose is the amount of the degree of electron beam irradiation. So it's often defined by the irradiation of beam energy per unit area in a specimen. And electron dose, generally represented by coulomb per square centimeter or charge density, often it is described as electrons per unit area, electrons per square angstrom, or electron per square nanometer. Now, if you really want to make a good image of free of statistical noise at especially high magnification, you need 1 coulomb per square meter of electron beam dose. It accounts for 6 into 10 raised to 4 electrons per square nanometer. Now let us consider what happens under these conditions for simple organic molecule. Most of the organic molecules, even some of the metal particles, can be destroyed even at 10 raised to minus 2. That is a 2 order of magnitude lower dose that is actually needed for a good image formation. Unfortunately, only a few compounds can be observed at the doses of the order of a few coulomb per square centimeter. What I'm going to show you today is a study of gold nanoparticle and done by a brilliant doctoral student, Abid Zulfikar, using in situ solid state TEM imaging. In this particular case, we study two types of nanoparticle. Both are gold nanoparticle of different size. Then we use two electron beam dose rate. Here, not just the dose, we also look at the dose rate here. And we consider this as a low and high dose rate. The nanoparticles average size of 6 nanometer and 11 nanometer with small and large size respectively. The very first thing that we are interested is look at how nanoparticle, this particular particle under beam irradiation for 1 hour undergoes transformation. Here is the real time, which has speeded up to 120 times than the original video. You see some of the structural reorganization going on, where the particle is not destroyed, but it has structurally reorganized. Here is a snapshot at certain time. Initially, particle will look like and what happened after 40 minutes and 63 minutes. What we observe throughout this video is a particle which is icosahedral, which becomes a twinned structure, transform into a decahedral structure, and then ultimately result into an elongated structure, and we can see two twin boundaries. Why this kind of structure happened throughout this transformation in this video, we have seen continuous diffusion and redistribution of atom density. What does this say for us is the time a TEM can be used to transform structures under continuous electron beam irradiation without destroying the particles at room temperature. Now, what happens if you take slightly larger particles? Here is a large particle which has a 5-fold symmetry, and then upon irradiation, we can notice that there is a structural transition to resulting in the 5-fold twin become D-twin structure. We will see the twin interfaces now migrates to the boundaries of these structures. What happens here is the surface diffusion of atoms when we go from all the way from 5-fold twin to D-twin structure here. Again, it involves surface diffusion, and final structure what we see is a truncated octahedron with a single twin boundary. So the size, depending on the size, we can transform into different final structure of the particles simply by irradiating beam. Now what happens if two nanoparticles are placed very close together and they are in mutual contact or spatially isolated? Here are two small size particles. And what you see initially, the particles do not change, but they start to come together and they start to merge. There's a neck formation, and ultimately it elongates, forms a rod-like structure. Finally, it transforms into a one particles, a larger particles. And this process is called coalescence. And we can take the snapshot and we can visualize how the structural transformation happens. Now the question is that why these particles come together and form a new structure. It's partly because of the high surface energy of the smaller particles, and they want to reduce the interaction, reduce the surface energy by interacting and becoming a larger particles. Now, what happens if you look at the two large particles? And here's a movie. It's not complete, but still shows there is a first two particles which have five-fold twin structure. They first come closer together. However, they do not merge so easily like the small particle, and that is partly because they have to perfectly align their lattice. So initially, these particles have a lattice mismatch and they reorient and they reduce this mismatch, and eventually we get, even after 1 hour, incomplete structure, rod-like structure formation. That's because the surface energy of larger particles are much lower than the smaller particles. And during this time, there can be a redistribution, and that is why this kind of incomplete structure formation, unlike the small particles. We can also see here individual atom diffusion. We can track how the atoms diffuse during this coalescence process here, and it's possible to track surface diffusion. Now, one of the important factors when particles come together is monitoring the neck growth rate. And this helps us to also understand the mechanism behind the coalescence or particle merging. Here we saw that how at low dose with the uh, violet colored graph, and with the two orange and yellow shows high doses. And we can determine the neck growth rate into a power law where K is constant and A refers to the order of the power in the change of the neck radius. That also helps us to understand how it is the mechanism behind this merging or coalescence of the particles. Typically in the literature, based on quantum mechanical calculation, A is between 1/6 to 1/7. However, what we found is that the value in our data for A is between 1/4 to 1/3, and it is significantly different from the prediction suggested by classical quantum theory in all observation for larger particles, ceramics, and microparticles for coalescence. However, one of the literature by McCarthy et al. Shows that if the mechanism of coalescence is facet-mediated surface diffusion, then the value can go 1.6 to 1.7 to 1/3, exactly what we observe. So our experimentally determined value of A therefore suggests that we have a surface diffusion. And we can also look at how the coalescence is formed by looking at the overall length of these particles and the diameter of the particles and the aspect ratio of these particles. And we can clearly see that in this case we saw two different particles and high and low dose, and there is a clear change in the aspect ratio and the overall diameter of the particles. For large size NPs, we can see there is little or no change in the aspect ratio, diameter, or the length of the particle because of the reason that there is an incomplete coalescence and it results in a new rod-like structure. Now, we have seen the individual particles undergo structural reorganization, and there can be coalescence between particles. But are there more such phenomena which are not well studied? Here are two particles, small sized gold nanoparticle, and they are very close in proximity. Upon irradiation of electron beam dose, we start to see they are not attracting, instead, they start to repel each other. And they repel to an extent from 0.9 nanometer interparticle edge to edge distance, and all the way now they go up to 17.8 nanometer under low dose. And here is the difference. Rather interestingly, this repulsion is much higher when we use low dose rate. However, at high dose rate, the repulsion is much smaller. Now the question comes, why this slow rate of repulsion happens at a higher dose? One possible explanation is that in the low dose, there is enough time to charge redistribution around these particles. When there is a charge redistribution happens, there's enough multipole charges are generated, which makes the particles repel. At higher dose, this redistribution is much slower and there is not enough time, high dose rate. Therefore, the multipoles may not be fully as in the case of low dose. Therefore, the effective repulsion is much lower. Now we can think what happens in the case of larger nanoparticles. Particularly here, we do observe repulsion, but more interestingly, one of the example what we say, initially there is an attraction, the two particles come together. However, after a certain time, these particles start to repel. And what we call it as sequential attraction and repulsion, this is something new that has not been fully studied before or observed. If you consider here, initially particles are about 2.9 nanometer. And then the particle distance decreases to 1.2 nanometer, and eventually we see the two particles repel up to 11.1 nanometer. Then why this such kind of attraction followed by repulsion happens? However, this requires much more explanation and computation simulation. But we can say that because in the beginning there is some charge redistribution, but continued electron dose, the charge redistribution becomes maybe less effective. We can also look at here what happens to a large particle. We can see a clear repulsion, whereas when we have high dose in large scale nanoparticles, we see first attraction followed by rapid repulsion. And why this kind of observations or behavior are important? What I have shown you now is not just looking at the particle, it actually electron beam mediated manipulating of nanoparticle, manipulating the structure, manipulating a new coalesced structure formation, manipulating their distance, controlling precisely how you can place two nanoparticles. Such understanding are useful in a multiple way. First, how extreme conditions such as radiation, temperature affect two nanoparticle placed together. Important for future nano-devices which are necessary for high temperature or radiation or pressure condition performance. Secondly, it provides us a new scientific insights. What is the effect of beam and how the particles behave under this? So manipulation as well as understanding the fundamental properties of beam nanoparticle interaction. In summary, I would say that metal nanoparticles are highly sensitive to high energy electron beam, which is in fact known. We can utilize today electron beam to precisely manipulate the nanoparticles. We can achieve new morphological features and optoelectronic properties. Materials and design for extreme environment conditions are possible if we are able to identify the correct